Hello everybody, what is up? Welcome back to the Bio Bulletin, the #1 YouTube channel helping teenagers like you and I learn about bio concepts. Isn't that so cool that we help each other? I think that's so cool. It astounds me that we all support each other. Anyway, if you're new here, hi, I'm Jada. If you're not new here, hi, I'm Jada. You probably know me as that weirdly obsessive science chick on YouTube that does biochem and extension science. It sounds so gross to some people, but I love it. Anyway, on this week's episode of the Bio Bulletin, we're going to be looking at biotechnologies. I'm sure you've all heard of them, they're in the news a lot. In particular, we're going to be looking at CRISPR this week. What is it? Are there ethical considerations? Will it impact on biodiversity? How much will it cost? Will society push back against it? And does it align with religious beliefs? Now, before we get started, quick disclaimer, the video may jump around to a few different locations today. It's just because I wasn't able to film the whole video in one sitting like I usually am, but I didn't want to miss an episode of the Bio Bulletin. I couldn't let you guys down like that. Now, before we get into all the juicy stuff that I know you guys are waiting for, let's hit the streets in Tina the Teeter to see what some other teenagers think CRISPR is. Alrighty, all ready to go. What is CRISPR?
No idea. Absolutely no idea.
Okay.
Okay, I have absolutely no idea what CRISPR is.
What is CRISPR?
CRISPR?
Yes, CRISPR. What is CRISPR?
I have no clue, honestly.
Okay. What is
Hello.
CRISPR?
Huh? Thing in the fridge?
The word sounds familiar. Maybe I learned about it when I was at school.
What is CRISPR?
I wouldn't have a clue.
No?
No. Nothing.
CRISPR?
Yes.
I feel like I've heard of that.
Yeah.
I just remember what it is.
CRISPR, it sounds like some sort of Wilbur variant word.
But it's giving me the vibe of robots. The robots that do surgeries. Don't know what they're called, but that's what I'm gonna guess. It's related to robots in medical situations.
Fries that are really crunchy.
Since none of them knew what CRISPR was, I'm assuming you guys don't either. So let's get into it. This is juicy stuff. It's exciting stuff. So CRISPR, or CRISPR-Cas9, stands for Clustered Regularly Interspaced Short Palindromic Repeats, with Cas9 standing for CRISPR-associated protein number 9. That probably still makes absolutely no sense to you guys. It didn't make sense to me at the start either. But in a nutshell, CRISPR is a biotechnology that allows for genome editing by drawing upon a naturally occurring genome editing tool that's found in bacteria. The name itself refers to the unique organizations of short, partially palindromic, which is just a fancy word for repeating or recurring, DNA sequences that are repeated in the genomes of bacteria and other microorganisms. CRISPR, before its use as a biotechnology, was actually part of the immune system of bacteria. It was the way in which bacteria recognized viruses and would attack them. It occurred through the Cas9 protein, copying a tiny little part of DNA from a previous virus and retaining the specific parts of the DNA. This meant that next time a similar virus attacked the bacteria, the CRISPR process would immediately recognize that virus and attack it. How cool is that? However, once this system was discovered by scientists, it was realised that the natural immunity system could actually be used to edit, remove, and replace genes in other organisms. The biotechnology allows researchers to easily alter and modify DNA sequences far easier than any other gene editing tools out there at the moment. CRISPR also has a wide variety of potential applications spanning across many, many fields, such as correcting defects, enhancing crops, and treating and preventing diseases. You can see why this is such an important biotechnology. Now, before we get into all the juicy stuff, the controversial stuff, the ethics, the applications of CRISPR, the benefits and drawbacks, the stuff I know you're all looking forward to hearing about, I'm going to give you a really quick overview of exactly how CRISPR works, so that if you ever need to talk about CRISPR at any point in your life, if it's in a bio class, could get asked questions about in the HSC since it comes under biotechnologies. If you need it at uni in the future, if you're going to study biology or biotechnologies or medicine, you'll understand how CRISPR works. So essentially, there are two elements to CRISPR, the Cas9 protein and the guide RNA, or gRNA, as you may see it in some places. Before the process of using CRISPR can even begin, the scientist has to decide if they want to disable a gene, insert a new gene, or repair a gene. Now, the process of CRISPR starts with a strand of RNA in the nucleus of the cell being edited, which is moved to the part of the nucleus where the editing is going to take place. This RNA is now known as guide RNA, or gRNA, as you may see it in some places. This gRNA then guides the Cas9 protein to the exact location in the DNA of the cell which will be edited. You can see why it's called guide RNA now. The Cas9 protein attaches it to the DNA at this point and unwinds the double helix shape, allowing the gRNA to pair up with the targeted section of DNA. The Cas9 protein will then snip the DNA at this location, creating a break in both strands of DNA. Since the cell senses a problem, it will immediately try to fix the break in the DNA. Fixing this break in the DNA may disable a gene, fix a mistake in the gene that has occurred during cell division, or scientists can use this opportunity to insert a new gene into the cell's DNA. So, now that you understand how CRISPR works, we can get into the juicy stuff.
Okay, Bio Bulletin, now it's time to get into the benefits and the drawbacks of CRISPR. Is it good? Is it bad? I personally think it's probably the best biotechnology that has come out of the world so far. It's going to revolutionize the way we edit genes, but let's get started. So CRISPR is really simple to use once it's set up. Once you have the CRISPR system all set up, the only thing you have to do is make slight modifications to the system in order to target different regions of the gene you want to edit. Then all you have to do is design and order your new guide RNAs, which can then be introduced into your already up and running system. So it's very, very easy to use in comparison to other genome editing systems we have out there at the moment. The number of publications using the technology has also skyrocketed, with more than 15,000 articles using CRISPR listed in PubMed alone. It has very quickly become a tried and true tested genome editing tool for a reason, which is it works. It's a simple yet effective way to investigate the function of your gene or genetic region. Clearly, because there are so many different publications that have all tested CRISPR, there's a reason we're investing time and money into this technology. It's a good technology. There has also been a surplus of research into how to make CRISPR more accurate. The research has surrounded using new amino acids in the Cas9 protein, changing its shape as a result, with one particular new variant of Cas9 called Cas9HF1 having an accuracy rating of over 99.9%. This is an insane accuracy rating. The fact that this protein has the ability to give the CRISPR system a 99.9% accuracy rating is unbelievable for a biotechnology that's as new as CRISPR is in comparison to other biotechnologies out there. As much as I love CRISPR, I could sit here and talk about it for days and days, I can't ignore the drawbacks. However, I can assure you right now, these drawbacks are only temporary, and with more research and advancements in the technology, they will be fixed. If they can't be fixed, there's just minor inconveniences in terms of all the benefits we get from using CRISPR. So the number one drawback is that the initial setup of a working and functioning CRISPR system is really quite time consuming. The setting up process, if your lab doesn't already have an established technique for genome editing, can be really challenging. However, once set up, CRISPR is such a simple and fast to use genome editing system, which easily makes it the best technology available for this purpose. There are also issues surrounding the techniques currently being used to improve the editing efficiency of CRISPR. with the editing efficiency referring to the percentage of cells that have been successfully edited. Now, this is not to be confused with accuracy, because efficiency refers to the productivity of the system, not how accurate it can make changes. An editing efficiency of less than 100% is by no means a disaster. It just means that the results have to be interpreted a little bit more carefully, which is currently where CRISPR is sitting at. The most common methods that are being used to improve the efficiency of the CRISPR system is to put markers in the Cas9 proteins, which could be a virus designed for gene expression. And the other technique is to select the population of cells that have successfully been infected with free nucleic acid. However, these approaches have two main potential fatal flaws, to be dramatic. The cells may no longer behave normally in the presence of some antibiotics, and if these successful cells do not readily divide or expand, then scientists may have to resort to a subpopulation of cells, which will limit the number of cells they have to work with. However, as I mentioned earlier, an efficiency of 100% is not vital to something being considered successful, and CRISPR is still evolving almost every day, which I believe still makes it a worthy technology to be backing. There are also concerns about whether CRISPR is specific in practice, which it is not. It can create mutations elsewhere in the genome, which are known as off-target mutations. These off-target mutations are completely random and can influence other genes or regions of the genome. They can be reduced by using a modified version of Cas9 known as Cas9 nickase, which just creates a little nick in one strand of DNA instead of actually breaking both strands like the CRISPR-Cas9 system does. However, even with this, you can still never be 100% confident that there aren't any off-target effects in a cell that has been edited using CRISPR, with the consequences potentially being catastrophic. However, like I keep mentioning, CRISPR is constantly being tested and worked on by scientists all around the world, and in the near future there will definitely be a CRISPR system that produces little to no off-target mutations. Now, the only drawback that currently is pretty dismal to me is that the success rate of CRISPR is only 1%, which essentially means there's only a 1 in 100 chance of CRISPR actually working for a scientist, which doesn't sound real good. But I guess my only comeback to that is, like I keep saying, that in the future the success rate will increase. With more advancements, more scientists working on it, this number will definitely increase in the future. While I'm waiting to interview some more teenagers for some upcoming little interview segments, let's go through the past, current and future uses of CRISPR. I've got a whole list of them up on my little apple juice laptop right here, so we're going to go through them and see what we think. So one of the major developments in the past with CRISPR was the development of what is called the Arctic apple. Now, the Arctic apple is an apple that actually doesn't brown, which I think that's really cool. They created this apple by blocking the enzymes that cause browning of food. Once it was confirmed that the saplings of the apples that they inserted the gene to block these enzymes into had actually accepted the gene, they were allowed to grow and produce the Arctic apple, which that's pretty cool. Like an apple that doesn't brown. So these apples are actually able to be purchased at select cities in the United States in little bags of sliced pieces, since one variant, the Arctic Golden Apple, has received approval from the Food and Drug Administration. Let's just talk about that for a second and how that could completely change our agricultural crop industry. If we can develop foods like fruits and vegetables and other types of food that aren't going to go bad, as we call them, like an apple browning, or just fruits and vegetables, they go bad after a little while. But if we can prevent that from happening, the drastic increase we could see in the supply of food in terms of crops would be immense. Like, that would hugely increase food supply in the world, since food, when it's being like transported all over the world, that's not going to survive a lot of the trips, or once it's in that country, it's not going to last on the shelves very long. So I think that this development and use of CRISPR, if it's applied to more agricultural industries, could definitely change the world for the better. Another use of CRISPR is that they've been able to create almost like a little DNA tape recorder, where Harvard University managed to create what's called CAMERA, which stands for, sorry, I'm looking at my laptop, this is hard to remember off by heart, CRISPR mediated analog multi-event recording apparatus. That just sounds like a whole bunch of words. But essentially, they manipulated CRISPR so that the guiding RNA will only take action after a trigger. And in doing this, the team at Harvard University created a tool that records the events in the lifetime of a cell. This camera system has been proven to work both in bacterial and human cells. That's a huge development. And this camera system now has the potential to be used for detecting environmental pollutants or tracking the signals that cause stem cells to grow into muscle cells, nerve cells, and other types of cells. Again, the implications of this use of CRISPR are insane. The fact that we can now change cells to be able to track all the events in its lifetime, and we can even use it to track environmental pollutants. Like, that research could be very important in terms of how climate change and pollution in the air affects the life of a cell. That research could be so useful to some scientists that are studying climate change and how it's impacting the world. And being able to track the signals that cause stem cells to grow into different types of cells, that's something that scientists have been wanting to work out for years. How one stem cell, which is a cell that can grow into anything, knows exactly how to turn into a specific type of cell. Like, to be able to work that out using CRISPR would be so amazing for the scientific community. So currently, in terms of the use of CRISPR, there are trials in China that are looking at CRISPR as a potential for cancer treatment. The patients have an advanced cancer of the esophagus and have what are called T cells, or they are white blood cells that are necessary in the immune system. And CRISPR is being used to remove the genes that code for a protein in these cells that some tumours use to grow. Now, these modified cells without that particular gene are then infused back into the patient, and hopefully they'll have a higher capacity to attack the cancer cells. At this point in time, 86 different people have been treated using this CRISPR therapy to get rid of the cancer, which I have no words. I'm at a loss of words for how amazing that is. CRISPR being used to treat cancer. You know, our only hopes for treating cancer at the moment are different therapies that are harmful to other parts of our body. Like, radiation therapy can have long-term consequences. So if we can use a technology like CRISPR to treat such a detrimental disease to people such as cancer with minimal side effects, and it's been proven to work, that would change the whole game in medicine. Like, people have been working for so long to find a treatment for cancer, and it seems like we may have just found one with CRISPR. CRISPR has also been used to treat another disease, which is muscular dystrophy in dogs. Muscular dystrophy is caused by a mutation in the gene responsible for producing a protein called dystrophin, which is needed in the body. but the mutation means that the muscles are weak and don't work properly. Typically, this disease is actually fatal because of the consequences it has on the body. The technology CRISPR was able to restore dystrophin in both heart and muscle tissue by up to 92%. These results are astounding and give way to the potential of using CRISPR to treat muscular dystrophy in the future. Which, like I keep saying, this is crazy stuff. Crazy stuff that this little old technology called CRISPR has so many different uses. Like, I don't understand why more people aren't talking about how cool this technology is. In terms of the future, this is where things get a little controversial. So, CRISPR is going to potentially be used for organ transplantation. Yes. Specifically, xenotransplantation, which is a procedure that involves either transplantation, infusion, or implantation into a human of tissues, organs, or living cells from an animal source. You heard me right. I said things were going to get controversial here. It is a growing problem that there is a shortage of organs in the world for transplantations. There are far too many people that need an organ transplantation compared to the number of organs we have available. It is suggested that using CRISPR, we could grow human organs in domesticated pigs. Yes. We're going to grow human organs in domesticated pigs in the future. There are still issues that scientists need to resolve before the possibility can become a reality, because there are obviously risks of viral infection and rejection. This potential implication of CRISPR in the future is going to ruffle a few feathers. People are not going to be happy because you're essentially using animals for human benefit. We are giving their lives no meaning other than to grow organs for humans, which, that's pretty bad. It's pretty bad when you look at it that way. It does not sound good at all. It goes against a lot of people's values and beliefs, and yeah, I can't see a lot of people agreeing with this use. Personally, I think that it will be beneficial because there's such a shortage of organs for medical purposes and transplantations at the moment. We are losing way too many humans' lives due to a global shortage of organs. And I believe that it would not take a huge population of pigs growing human organs to solve this problem. There are still currently issues, like I've mentioned, but hopefully once those issues have been ironed out, this technology could become a reality. Another potential use of CRISPR in the future is to revive extinct animals. If scientists are able to construct the genome of an extinct species of animal, then those genes could be inserted into a close relative in order to essentially bring that species back to life. In particular, they're looking at the woolly mammoth, using samples that have been preserved in the Arctic permafrost of the woolly, samples from the woolly mammoth that were preserved in the Arctic permafrost. Scientists were able to construct their whole genome and are looking at inserting the genes into the Asian elephant, which share 99.96% of their DNA, which makes them a pretty ideal starting point. So the team wants to use CRISPR to insert genes that will code for dense hair, extra fat, improved oxygen carrying abilities, essentially the traits needed to live at approximately -40 degrees Celsius, just like the woolly mammoths did. These hybrids will hopefully look and behave exactly like the woolly mammoth did. I think that is so amazing that we're going to be able to bring back extinct species. Like, obviously we won't bring back anything that is very, very dangerous to us. Scientists are going to be careful with what they do, but the thought of bringing back an extinct species, like, does that not get you excited? Because that gets me excited. Like, that's sick. Do you think there are any ethical concerns?
I think the whole like changing the body, like usually when it's appearance it's not like that drastic, but like the whole like changing this like structure of the body,
Yeah.
it's a bit, yeah.
Do you think it will impact on biodiversity, with biodiversity being variability in organisms?
I think it could probably create more diversity, because would there be like different combinations?
Yeah, there could be.
Yeah, so it'd be like, it'd probably make it more diverse in a sense.
Thank you. I'll double check that with you. Do you think that it raises any ethical concerns? Do you think there's any ethical concerns?
Yeah, obviously, with like farming and stuff like that.
Yeah.
And other countries who might be producing stuff, and then other countries who aren't good at producing that same type of thing are now going to edit stuff and make it better and worse for other countries.
I think so.
Do you,
Stuff
so, do you think there are ethical issues in terms of CRISPR?
There could be mutations. Any like after results could be like, yeah, it could start maybe a disease. I don't know.
I think that the only reason that CRISPR wouldn't be ethical, but it's something for a lot of like things, even makeup brands, it would just be like the animal testing, which sucks.
I don't know. Maybe. Yeah, sure. We'll go with yes.
Do you think there are ethical considerations?
Yes, definitely.
It is now time to talk about the ethical considerations. My personal favourite topic. Here in the biology field, we sure love to talk about ethics, so let's just get right into it. The number one ethical concern for CRISPR is its usage to genetically modify human germline cells and embryos. This is always an ethical concern that comes up time and time again with biotechnologies, is using it to edit human cells. Now, there's not a problem with using it to edit body cells or somatic cells if you want to be a little bit fancy, which this had been occurring up until 2015. Totally fine. But some Chinese scientists decided to edit human germline cells using CRISPR, which raised new moral, social, and ethical issues, as you'd expect. Now, because CRISPR is known for some off-target mutations, this was a huge concern with editing human germline cells, because these undesirable changes could be transferred to future generations, since germline cells are sex cells or gametes. It was actually found that there were lots of off-target mutations, and the study done by these Chinese scientists was shut down immediately. Researchers have also recently found out that off-target mutations are much more common in embryos rather than fully grown humans and mice, which that's a massive concern if we're looking at ethics. Another concern is equity surrounding CRISPR-Cas9. There's concerns that while it's relatively affordable in comparison to other genome editing tools, CRISPR will only be accessible to the wealthy. This prospect is suggested to only increase the already existing differences in access to healthcare as well as other resources between the rich and the poor, with the extreme version of this ethical concern being new classes that are defined by the quality of their CRISPR engineered genome. That's a pretty scary thought. The idea of different groups of individuals, like the rich, the working class, and the poor, but instead it's the high quality CRISPR engineered genome and the low quality. That's a pretty scary thought. There are absolutely some positives in terms of ethics surrounding CRISPR, with the possibility of a disease-free world, which I'm sure after COVID-19, we'd all be cheering at that idea. Like, that sounds so good to us, but it particularly applies to genetic mutations, not infectious diseases. But
There's over 10,000 different diseases that are caused by a genetic mutation in a single gene, with even more caused by genetic mutations in multiple genes. But CRISPR has the possibility to switch these genes off, leading to a disease-free world. That'd be pretty cool, considering genetic mutations can cause deaths and these diseases have such a high prevalence in the world at the moment. But on the flip side, is the world really ready for a disease-free world? Like, if we especially look at plants, if CRISPR switches off genes and edits out diseases that are caused by mutations, there's the possibility that species may not be able to survive with the constantly changing environment due to climate change. If CRISPR edits out a mutation in a plant that causes what we currently see as a disease, but in the future with climate change, all the climate changes, what if that mutation would have actually meant that species could have survived? So that's something else we have to think about if we're looking at a disease-free world. So, in terms of humans, we might see it as a good thing, but if we're looking at plants, is it really all that great of a thing? Because that could have a pretty detrimental effect on the food supply of the world if suddenly one of the most widely used crops in the world dies out due to not having a genetic mutation that would have enabled its survival.
Do you think it will impact on biodiversity, with biodiversity being variability in organisms?
I think it could probably create more diversity, because would there be like different combinations?
Yeah.
So it'd probably make it more diverse in a sense.
And do you think it will impact on biodiversity, with that being variability in organisms?
Yes.
Yes. And do you think it will impact on biodiversity, with that being variability in organisms?
Yeah, because, you know, evolution, stuff like that, if you're making something consistently the same, it won't really ever kind of be different. It'll just be the same forever.
I reckon it'll decrease it. I reckon they'll there's gonna be some sort of mutation or backfire from it, and in result of that, they'll probably slowly get wiped.
I don't think it would affect it majorly, but I think it could.
Do you think it will impact on biodiversity, with that being variability in organisms?
It'll definitely increase biodiversity. There could obviously come some issues with chopping and changing.
It is time to talk about biodiversity. Every biologist loves this topic, or consideration when it comes to biotechnologies. If you talk to any biologist, I think, and mention biodiversity in relation to a biotechnology, I can almost guarantee you that they will not shut up about it. They always seem to have arguments for or against it. So let's look at some arguments for and against it. However, would just like to give a heads up, CRISPR generally has pretty positive effects in terms of biodiversity. Like, it's such a great biotechnology that it honestly improves biodiversity for the most part, with biodiversity being the variability in organisms in ecosystems. So first up, we have that CRISPR may be able to help secure biodiversity in terms of food security. That may come as a shock to you all, that biotechnology increasing biodiversity, like most of them decrease it, but CRISPR just isn't like the other biotechnologies. So over the past 12,000 years, humans have been selectively breeding plants with characteristics that are more favourable, creating domesticated crops. However, in doing this, the biodiversity has been significantly reduced. And with this, farming has become much more predictable with ideal results every time. However, with climate change increasing both variability and unpredictability in agriculture, the plant's already low genetic diversity is going to make them susceptible to a further loss due to plants being unable to cope with the fluctuating conditions. However, CRISPR, like it always is here to save the day with a solution to maintain biodiversity. The prospect of inserting genes to make plants better suited to the changing climate conditions through the use of CRISPR, pretty exciting. An example of this was scientists were able to insert genes into wild tomatoes that make them grow exactly the same as the domesticated crop. However, it's completely genetically diverse compared to the domesticated tomato. This breakthrough in science provides an opportunity for farmers to have crops that will be better suited to climatic challenges that may arise in the future. This just increases biodiversity, like having crops that all grow the same but are genetically different, which means that if conditions suddenly change, there'll still be crops that survive. We're not going to lose any biodiversity. If anything, CRISPR is improving biodiversity. It should not be associated with any of those other genome editing technologies that decrease biodiversity. CRISPR is also going to bring back biodiversity in terms of coral. As we all know, coral is significantly diminishing due to coral bleaching, which has happened due to global warming. Now, gene editing through CRISPR is seen as a possibility to bring biodiversity back amongst coral, with the intent being to use CRISPR in laboratory settings to gain an understanding of which genes in coral are important for handling stress. The information can then be used as a blueprint for editing coral genes in the future for conservation. Or it can also be used to study coral populations that are more tolerant to warmer waters, which could then in turn be used in conservation efforts. I think this sounds pretty good. Like, we all know that coral bleaching is a massive problem. All the corals in the world are dying, especially in the Great Barrier Reef, due to global warming, which was caused by us. So if we can bring back that diversity through another technology, that would just be so good. Like, you can tell I'm really passionate about this stuff. Like, that would be so good if we can bring back the biodiversity in coral that we took away. I think I'll just balance everything out. Like, CRISPR is just so amazing. It is definitely going to increase biodiversity. The only reason it may decrease biodiversity is that if organisms that have had CRISPR edited genes somehow escape the facilities that they're being kept in, they can negatively impact ecosystems, as they may increase competition with other native organisms. But this is not a problem because CRISPR edited organisms are currently kept very secure in labs. So, in terms of biodiversity, CRISPR is great. I'm all for CRISPR. Like, those two examples that we just talked about clearly show how CRISPR is only going to increase biodiversity, which is definitely needed in a world where climate change is threatening the loss of huge amounts of biodiversity.
Do you think CRISPR is cheap? Doesn't sound like it would be. It sounds like really expensive.
Yes.
Do you think it's cheap?
No.
No. Do you think it's cheap?
No. Oh, the development probably isn't, but the use of it probably is somewhat cheaper, but not that cheap, I'd imagine.
Do you think CRISPR is cheap?
No, absolutely not.
I think far into the future, obviously, if you can stop somebody from getting cancer or giving them that gene that could possibly give them cancer, it's going to save them a lot of money.
Do you think CRISPR is cheap?
I don't know how much it is.
This is probably a question you've all been waiting for. Like, biotechnologies, as we know from past Bio Bulletin episodes, tend to be pretty expensive. But is CRISPR affordable? Let's find out. So, most genetic editing tools cost thousands and thousands of dollars, which is just unreasonable to think that they could be implemented. But on the other hand, CRISPR's relatively cheap, making it very feasible to implement in the world. Like, this is all subject to change in the future depending on the different applications of CRISPR, but currently, scientists can create a short RNA template using some free software over the course of a few days, and then they can purchase a DNA starter kit for just $65 US or about 85 Australian dollars, and shipping. That's it. That's how much CRISPR costs at the moment, which is extremely low in comparison to other gene editing tools that cost upwards of $1,000. Like, I don't know why scientists are even using other gene editing technologies at this point in time. Like, $65 US. $65 US compared to 1,000. Over 1,000.
why are we still using other editing technologies? Like, I know CRISPR isn't perfected yet, but once it's perfected, there is no use in using other gene editing technologies when we could be using CRISPR for the low cost of $65. Yeah. Do you need to make a quick little point that CRISPR could increase in price if it's implemented as a type of gene therapy? Because gene therapies tend to cost about 6 figures or upwards, with gene therapies such as Glybera, which was made by the Dutch company Unicure. This gene therapy costs $1.4 million per patient. That's very expensive. Doesn't sound anything like the $65 we were just talking about. So CRISPR, if it's implemented as a gene therapy, we can assume the price is going to increase. In fact, a gene editing company focused on developing CRISPR related treatments called Adidas has just signed a $737 million contract with Juno Therapeutics, which is an American biopharmaceutical company, to combine CRISPR with other tactics in order to treat cancer. However, if CRISPR as a gene therapy becomes more widely available with more uses and it's more accessible, the price will decrease. Any biotechnology or medical sort of application, if it's widely used, widely available, the price generally decreases because everybody can use it. They don't have to charge such a high price for just a few people to use it. So, all the more reason to implement CRISPR as a more widely used biotechnology, aren't I right?
Do you think society will push back against this technology?
I think at first they might be a bit hesitant to like implement it, but once they see like the benefits of like the eventual benefits, they'll be like, yeah, let's go for it.
Do you think there will be pushback from society?
Yes, definitely.
I don't think it would cause a setback from society. It could. I really have no idea. Maybe, maybe not.
Do you think there'll be pushback from society?
Yes and no. Like, there'll be yes because like they don't want to eat genetically modified foods or have products that have ethical downsides, but yes, because it might make things more accessible or, you know, better quality.
Do you think there will be pushback from society?
Yes, there will be. There'll be all those like anti-needle people.
Do you think society will push back against it?
No. I reckon the good outweighs the bad.
At this point in the video, I'm going to assume most of us are all for CRISPR. Like, I don't understand why you wouldn't be against it. With all the amazing applications that CRISPR has and what it can do for the future in terms of biodiversity, I don't understand why you'd be against it. But there's some people in society that are, so it's important to understand if there's going to be pushback from society against CRISPR. So that's what we're going to talk about right now. So currently, the national and international guidelines surrounding biotechnologies, including CRISPR, provide very little guidance and oversight for the applications of technologies, which in doing this, it doesn't really foster public trust in CRISPR and the safety of CRISPR-edited organisms. This just generates a public misunderstanding of CRISPR, which is going to hinder its advancement in the scientific world. However, if we create an ethical framework and some strict guidance that scientists have to adhere to in terms of the use of CRISPR and its applications, the public's trust in this technology will increase, reducing pushback from society. So if we can get that up and running, less pushback from society. Sounds pretty good to me. However, there are also increasing movements in society against scientific development, which could mean that they'll also push back against a technology like CRISPR. As we all know, the anti-vax movement is growing. Although this isn't a large percentage of the population, anti-vaxxers have increased their following by 7 to 8 million people in 2019, with 31 million people following anti-vaccination Facebook groups and 17 million people subscribing to similar accounts on YouTube. So these statistics I've just mentioned illustrate the rise in anti-vaxxers, as most of us call them, over the past 1 to 2 years. Now, as I've said,
with a world that has over 7 billion people, if a couple million are against scientific technology, it's not going to necessarily hinder the advancement of the technology too much. But this does illustrate that some people may push back against science because there's increasing movements against the development of scientific technologies, which it can be because of the future implications of the technology, some misunderstandings about the use of it. Like, we all know the misconception that vaccinations cause autism. So this could impact on CRISPR in terms of society pushing back, but it's a small percentage of people. So I think CRISPR's still in the clear. There's another phenomenon that we also need to consider in terms of society pushing back. There is another phenomenon that we need to consider when looking at society pushing back, which is the neophobia phenomenon. Now, neophobia is a fear of anything new, and it is typically manifested as an unwillingness to try new things. Now, CRISPR is a new technology. People with neophobia don't like new things. Don't think they're really going to pair well together because people are going to be afraid to step out of their comfort zone and try something new. This phenomenon has already had an effect in terms of pushback against other biotechnologies, such as genetically modified organisms, where a survey was conducted to see if people were willing to try lab-grown meat that was more sustainable and animal-friendly as an alternative to meat produced in the livestock industry. Now, because of this growing neophobia phenomenon, respondents showed a low level of acceptance in terms of the genetically modified meat, with majority preferring traditionally produced meat. This shows how, once more widely implemented, CRISPR could face pushback from people that suffer from neophobia. Again, like with the anti-vaxxers, this is probably just going to be a small group of people, and if CRISPR becomes even more widely implemented, the fear of a new technology may even decrease because it won't be new anymore. It'll be a pretty common technology. So I still don't think there's anything we have to worry about in terms of pushback from society, since it's only minorities that are going to threaten the advancement of CRISPR. But it seems like majority of society is pretty for using this technology to better our lives. Do you think it aligns with religious beliefs?
No, not really.
I think it aligns with religious beliefs. This is a good question for you.
No, I don't believe it aligns with religious beliefs because you are... How to phrase this? You are interfering with the creation of God and manipulating a living organism. Yes.
Do you think it aligns with religious beliefs?
Oh. I don't think it does to some extent, but it also doesn't, because like the religious belief is like God created you the way He wanted to create you and all that. But it could also align because it's helping out others and like you're seeing a need and doing something about it. Yeah.
Do you think it aligns with religious beliefs? Like Christianity?
I have no clue. Maybe not.
Do you think it aligns with religious beliefs?
I don't think it aligns, but I don't really think it goes against.
I think religion is a very different thing for like different people. I think, you know, there's religions that like won't like accept blood in medical instances. Oh my god, I can't say that word anyway. I think for certain religions, probably. For majority of religions, I don't think it would really matter.
When looking at any new biotechnology, religion is always something that has to be considered by scientists, by your everyday people, by just about anyone. So that's why we're going to touch on it now. Now, just before I get into the whole religious aspect and whether CRISPR aligns with religious beliefs in the world, I myself am atheist. So in saying that, I don't feel like it is right for me to provide my opinions in this section. I believe this section, it will be most respectful for me to just explain to you the viewpoints of particularly Christians, since they account for 2.38 billion people of the world that practice religions. So this section is going to be respectful towards their viewpoints and not about my own viewpoints. So the arguments made by Christians in terms of whether they agree or disagree with CRISPR surround the beginning of life, the creator-human relationship, the image of God, and stewardship. In terms of the beginning of life, they see it as the very beginning or inception, which is why arguments against CRISPR are made by Christians if it involves embryo-derived stem cells. Their belief is that living humans, including embryos, must be treated with respect, and they believe that the editing of genes in an embryonic stem cell is essentially killing a living human, which is fair to say given their beliefs. There's obviously also the question of humans controlling the natural world, which lines up with the creator-human relationship. In Christian beliefs, God is the creator and everything else is a creation of God, which should not be able to make any creations themselves. Therefore, the argument is made about whether genome editing through the use of CRISPR is a human role, or whether it's playing creator in colloquial terms. Some argue that gene editing oversteps the Christian ideas of human role, and particularly editing in germline cells fails to respect children as a gift from God. The image of God helps form the basis of another argument, both for and against gene editing. Man is considered to be made in the image of God, and therefore using CRISPR to change an individual's genetic structure is considered mistreatment of a gift. However, it's also argued that genetic editing can be a tool for people to fully express their identity. Religious bodies actually fully support the editing out of congenital diseases. These are diseases that are present from birth, such as Down syndrome, since these can negatively affect both the physical and mental identity of a person. And finally, the idea of stewardship, which is the job of supervising or taking care of others. This is a responsibility entrusted to humans by God, and therefore the idea of using CRISPR to edit the genes of humans raises the question of whether it's taking care of other humans or harming them. Genetic technologies tend to have a reliance on stem cells for research, and since this is viewed as harming or killing cells in the eyes of Christians, it positions genetic editing to go against stewardship. However, it can also be argued that genetic editing could increase the chance of survival for a fetus or human, which would then adhere to the idea of stewardship. Now, these religious viewpoints are integral to consider when looking at CRISPR and how they align and collide with the technology in order for us to have a full understanding of whether we should be for or against this technology, and to be more understanding of people that have different viewpoints to ours, especially if religion is an aspect of that. Okay, we've finally reached the end of this video. Now, this Bio Bulletin has been a long one, but it's because there is so much to be considered when looking at CRISPR. We've covered what it is, how it works, the benefits and drawbacks, its uses both in the past, currently, and the future, ethical considerations, biodiversity, how much it costs, pushback from society, and religious beliefs. That is a lot in one video. Now, after talking about all of that, hopefully you have come to decide whether you are for or against CRISPR. Let me know down in the comments below whether you think CRISPR is good or bad. Personally, I'm going to give you guys my final judgment. So, I am absolutely for CRISPR. I think that the idea of editing genes is definitely a good one in terms of where we're heading in the future with science and what we're hoping to be able to achieve. The way it works is really simple, since it is a naturally occurring immunity system in bacteria that we're just making use of for our own purposes. So it's pretty good in that way.
and all the benefits, obviously, of a version of the Cas9 enzyme that has a 99.9% accuracy. Like, even though there are some off-target mutations at the moment, these problems will diminish with further developments in CRISPR, and in no time, CRISPR is going to be this holy grail of a biotechnology, which it already kind of is. Like, it's being tested a lot. Some other reasons I am for CRISPR are that it's really, really cheap at the moment. Like, $65 is insanely cheap compared to other biotechnologies, and then once it's widely implemented, the price still shouldn't go up too much unless it only becomes available to a specific group of people. I do understand that there is going to be some pushback from society, but it is, in my opinion, that these groups are just minorities. They are not a big contributor in terms of the population of the world being over 7 billion people. Like, 20-30 million is not much in comparison. In terms of religious beliefs, like I said earlier, I'm not going to comment on my viewpoints, but since I am atheist, those viewpoints do not apply to me. So I am absolutely for CRISPR, and even more reasons to be for it. The implications it's going to have in the future. We're going to be able to cure cancer with CRISPR. Cure cancer. We've never had a treatment for cancer that can cure any type of cancer without any damage or long-term consequences on the body. So if we can do that with CRISPR, that is going to have insanely good implications in the future. And being able to prevent crops from being able to go bad, like an apple that doesn't brown, imagine how much that is going to increase our available food supply if we can edit crops so that they don't ever go bad. Like, that's going to work wonders, especially for third world countries where food is scarcely available. And to be able to treat other diseases such as muscular dystrophy, like, that's going to be sick. Like, CRISPR has the prospect to treat so many different diseases. Like, what was it, 10,000 different diseases caused by genetic mutations? If we can edit those out, and like, if we can edit out those mutations that are potentially deadly, that is going to be life-changing for some people. Again, there are also ethical considerations that we talked about, such as germline editing, which does raise a lot of questions, as well as creating a divide between the rich and the poor, with the extreme being classes determined by the quality of the genetic editing. Now, these are just a few ethical considerations. However, in terms of other biotechnologies, there are nowhere near as many ethical considerations for CRISPR. Especially when you look at biodiversity. CRISPR is only going to help the world. It is not going to decrease biodiversity. It is going to increase biodiversity. It is going to make sure we have higher food security, and it's going to increase the biodiversity that has been lost due to coral bleaching. So overall, my opinion, yes for CRISPR. It gets a double thumbs up from me. I'd give it a triple thumbs up if I could. I am 110% absolutely for the use of CRISPR. I think it is going to revolutionize the world. But I hope you enjoyed this week's episode of the Bio Bulletin. Be back next week for a new episode. Give me some suggestions on what you'd like to see in the comments below as well. Bye guys, have fun in bio. I'm definitely putting that in. Wait.
Wait, give us a second.
Good point.
Oh
I haven't
my
thought of
God.
that one. It's going to be $65.
What?
$65 USD plus shipping.
Plus shipping, you know, because you can actually
Yeah.
just ship a genome. So why do I not have blonde hair? Like,
Change their genetic makeup.
So I was close.
Close
Okay.
with the chips.
Yep.
Russ. You just keep clicking behind the video.
It's okay, Gabby said crunchy chips.